Once again, I would like to re-emphasize that if you do not understand anything, please raise your hand at that point. Don't wait for me to finish it, okay? And ask me the question, because by asking question, you're helping yourself and you're helping everyone else, and you're breaking the monotony of me speaking all the time. So please ask questions if you do not understand anything at any point, okay? And as I go along, I will show you a lot of things. These are pictures which have too much information. You don't need to know all the information. So I will tell you what are the important things, and all of you will get, Professor Venkatesh will send you the PDF files of these lectures, so you will have a copy of everything that I speak, okay? All right, so this is still an introductory lecture, okay? So as we go along, you will see that I will introduce many things and then I will go in more details as we go along, okay? So this will tell you the current landscape of therapies targeting many lethal cancers, precision medicine, small molecules to monoclonal antibodies. So the goal of this lecture is mainly to tell you about what were the traditional therapies for cancer, which we still use, but they were originally, they are still considered traditional because that was the first things we used to do long time ago. But now we have many new ways to treat cancer, okay, or stabilize cancer. So this is chemotherapy. We all know chemotherapy usually, at least in older times, chemotherapy was the main way. And do you understand what is chemotherapy? What is chemotherapy? Chemotherapy means you give those chemicals which stop cell division, okay? But what is the problem with chemotherapy? Every cell in our body is dividing, especially like, every single cell, right? So when you give chemotherapy, cancer cells are dividing very quickly, so they die, but the normal cells are also affected. That's why you see cancer patients, they lose hair, right? Because these cells are dividing very quickly. You may know that these stomach cells, all these things, they divide very quickly. Why? Because they are churning food, right? So they get damaged very quickly. So the stomach lining are replacing, making new cells all the time. So when you give chemotherapy, stomach is really upset because those cells are dying too, and people feel nausea and all the other things. You may have heard of this phrase that cancer will not kill me, but chemotherapy will kill me. So when the cancer becomes pretty bad, when the body is healthy, initially the person can take chemotherapy because body is healthy. But if you keep on giving chemotherapy, it is so toxic that the patient decides it's not worth taking chemotherapy. It's much better to just accept it and just, because it is so toxic, okay? But chemotherapy can still hopefully cure some cancers, which is not too spread out and not too aggressive, okay? So this is the oldest way of treating cancer. We still use chemotherapy, okay? Radiation therapy is that, suppose somebody has a tumor, there are two ways to give radiation. So somebody has a tumor, then you give radiation very, very specifically from an outside source to that location, and the idea is that you will kill the tumor cells. Or you can give radiation internally, I'll tell you about that also, okay? If you give it in a very localized manner, you will not destroy normal cells, okay? And then there is this targeted therapy. Targeted therapy is a big umbrella with many, many ways to target cancer, okay? So traditional methods of treating cancer are chemotherapy, radiation therapy, surgical therapy, okay? Yeah, surgery is obviously the first thing. If a cancer is localized, the first thing you do is surgery. You just remove it. And if you are very careful in removing it, and if it has not spread out, you can really basically cure cancer, okay? Many cancers, I know of a person, she had breast cancer when I was a child, okay? And she got her cancer removed, and she's still living today without any cancer, ever, okay? And there are many people I know back at Purdue who had breast cancer when they were young. And now they are in their late 70s, no sign of cancer for decades and decades. So sometimes surgery is very effective, and that is the best way. If we can surgically remove it, why expose normal cells to any pressure, right? So radiation therapy and chemotherapy, I already told you. So these are some of the therapies. There are so many therapies that I cannot classify it on one page, but these are some of the therapies. So immunotherapy is becoming very popular, and there are many different kinds of immunotherapy. So these are some of the immunotherapy, gene therapy, nanotechnology, cellular therapy. We will discuss all of these things, okay? So in this figure, it tells you surgeries was done in ancient times. Radiotherapy started in 1890s. It is still used present time. Cytotoxic chemotherapy actually started in 1940s. Okay, it is still used. Okay, molecular targeted therapy started in 1990s, and it is one of the most common way. Immunotherapy, it says 1990s, but it was not that popular in 1990 because, and even I know of a case, a person, he was my friend, he got a very rare, we both worked on cancer, and he was a fantastic scientist, and so he got a very rare cancer in one of the bile duct, and because we worked on cancer, we knew, what medicine. So his cancer was spreading very quickly. Somebody asked me today that, do you get warning? It depends on the kind of cancer. So he had no warning at all. He was exercising every single day, eating very healthy, but he still got cancer. So and it happened just 2-3 years ago, not very long ago. And here it says immunotherapy from 1990s to present. Venkatesh, you know that guy too. He got cancer very quickly. So immunotherapy was not approved for at that time. And so he said, I need immunotherapy. Okay. So he signed a lot of paperwork because, it was not FDA approved. So he signed a lot of paperwork. It took him 15 days, a month to get all the paperwork out of the way. Then he was given immunotherapy. It was too late for him. And from the day of diagnosis, he passed away within 6 months. It just spread out so fast. So immunotherapy is becoming very common these days. It was not even that common 3-4 years ago, and I know of people in our country here who are undergoing immunotherapy, and it is really helping them. So it's worldwide available now in all, in most of the countries, and it's a very effective therapy. So once again, so what is surgical theory? It's, you surgically remove it. Radiotherapy, you point radiation at the diseased part, and then chemotherapy is medicine. So chemotherapy and radiation, if you compare it, chemotherapy is a systemic therapy. What is the meaning of systemic? Every cell in the body is exposed to that therapy, right? So that is systemic. Systemic is good when cancer has spread out. You cannot go and remove cancer from one organ to the other organ. That's when you have to give systemic therapy. What is the downside of systemic therapy? Normal cells are also exposed to that really bad therapy. Okay? Radiation is still more localized. You can localize it to a specific part of the body. So that is good. But the worst part of cancer is that it becomes resistant to everything. It will become resistant to chemotherapy. It will become resistant to radiation also. So it's a constant changing of cancer cells. So there are two kinds of radiation therapy. One is called external beam radiation therapy, or ERBT. So in this we give, so even if the cancer is within some organ, it's not surface cancer, it is within some organ, but you're giving the radiation from outside. Okay, so this patient is given radiation at a specific part in the body. It's a high energy radiation, and tumor is somewhere in the stomach in this case here. And it is the most common form of radiation therapy. It uses a machine outside the body, and we don't care about the machine, what kind of machine it is. It is just giving high radiation energy, okay? And this Why this is used? It is used for many cancers like breast, prostate, lung, brain. So it's a very common kind of treatment for many different kinds of cancer. It can be curative. You can cure cancer by radiation, which is very good. Sometimes what you do, when a person has cancer, you remove it by surgery, but you cannot see every single cell. So you are afraid that there may be some cells which are still lingering around the place where you have surgically removed it. Now you shine radiation. If by any chance some cells are lingering around, they will die. Makes sense? So that is called adjuvant therapy after the surgery. Sometimes what you do, you give it before surgery. That is called neoadjuvant therapy. Like you are killing most of the things beforehand and then you are removing the tumor. That's neoadjuvant therapy. And palliative is that when you know you cannot cure the cancer, you are just relieving the symptom so that the person has better quality of life. Okay, like it is too far gone, but you're just helping the person. So it is used for many different kinds of things. So what are the advantages? You deliver very high dose to tumor cells. You don't damage the surrounding healthy tissues. Okay, so this is once again repeating the same thing. Okay, and then there is also internal radiation thing. So I thought I had a slide for the internal radiation thing also. Yeah, it is here, sorry, it is, I missed this part. Internal radiation therapy is that you actually place a catheter inside the body, and this one has radioactive source. So you actually put it right next to the tumor, and this is the radioactive source, and you are just killing that part of the tumor. So it's even better than external one, and this is even less damaging to the rest of the healthy tissues. This is also not that bad because it's not chemotherapy all body. It is still at localized point, but this is even more localized. Okay, but the hard part is to get inside the tumor. If it is very deep in the body, then it becomes a very invasive process to get it right at that stage. So that is the disadvantage. Okay. So like I said, it is very hard to categorize in one slide all different kinds of therapies which exist here. So in this one it tells you for cancer treatment in 2023, what are the most common ones? Gene therapy, chemotherapy, cell therapy, immunotherapy, targeted therapy, precision oncology. Oncology of course means cancer. Precision means like you are just killing the real culprit in cancer cells. So that is precision, radiation, and nanotechnology. Okay, so I will tell you, I have already told you about immunotherapy without telling you exactly what is going on in immunotherapy. What I told you, that in immunotherapy you are waking up the immune system, okay, so that they can recognize cancer cells and they can kill the cancer cells. Okay, and how, that I will tell you later. And gene therapy is that, suppose you know that this guy causes cancer because of this mutation, right? If you know, so if you correct that mutation, cancer should disappear, right? You are actually, or in other ways, suppose P53 is not present in cancer cells, you give them P53. P53 is tumor suppressor. That should fix the cancer pathways, right? That is gene therapy. You are actually adding the gene there, or you are correcting the gene there. How many of you know CRISPR? Probably all of you, right? And you know CRISPR can edit the gene. CRISPR can correct the gene, right? You can insert a new gene using CRISPR, right? You can remove a gene using CRISPR. So that is gene therapy. And CRISPR has made it very, very, very easy. So before CRISPR, you know how hard it was to remove some gene from an animal? It used to be a years and years long project, right? It was very hard to mutate a gene. It was very hard to remove a gene in animal models, right? CRISPR has made it a piece of cake to generate animals with correct genes, okay? So that is gene therapy. And targeted therapy, like I said, if I know HER2 is the culprit, I give the drug against HER2. That is targeted therapy. I know what is the problem and I target it to that problem and I give the therapy. Okay, so these are all of those things. Okay, so now I'm going to give you some examples of targeted therapy. So this cartoon is very good. This is a normal cells which has some cell receptors. Now imagine, so what happens in these cell receptors? They take growth factors. Growth factors turn on the signaling pathway and the cells divide, right? But what cancer cells do, they have too many receptors. More receptors, more signaling for growth, cancer, right? So if you know this cancer has too many receptors, now what you have to do, you have to inhibit these receptors to treat the cancer, right? So what you do, now you have to shut down these receptors so that they cannot bind the growth factor, and so there is no signaling. So very good example is that prostate cancer, they have androgen receptors, right? They bind androgen, correct? So what you do, you give anti-androgen therapy so that the molecules, they bind those receptors like that, but now they cannot bind androgen, right? So you are basically, the receptors are there, but they're ineffective. You can keep on adding androgens, but they cannot bind because the inhibitor is bound at the same site. It's the competitive inhibitor there, right? So androgens will make no difference to that, right? So that's what. So that is a targeted therapy. You are killing the receptors by binding it to some other thing. So there is no signaling going on there, okay? So this is just telling you the same, that the cancer cells have these signals, and you can actually address all of these problems using specific targets. We will not go into the details because that's huge signaling pathways. But basically, if you want to, so for example, this cellular metabolism is different between cancer cells and normal cells. So you know what is going on in cancer cells. Who asked me about Warburg effect? You asked me about Warburg effect, right? So Warburg effect also means aerobic glycolysis. Okay, so what is Warburg effect? So in Warburg effect, there was a scientist named Otto Warburg. Okay, so he was there in 1940s. So in normal cells, if you have oxygen, then glycolysis slows down. And why glycolysis slows down in the presence of oxygen? Because when you have oxygen, Krebs cycle is going on, right? Krebs cycle forms NADH and FADH2, and they go to electron transport chain, which uses oxygen. So the cells are generating so much ATP during Krebs cycle that they don't need glycolysis, right, to make ATP. So glycolysis slows down. So what Warburg saw, that cancer cells, even if you have oxygen, glycolysis is still upregulated. Okay, so that is why he termed it as Warburg effect or aerobic glycolysis. That means even when oxygen is present, glycolysis is happening. And why it is so at that time? So he said a very good thing, which is even true today, almost after 90 years, 85 years, right? It is absolutely true. Cancer cells, even if oxygen is present, glycolysis is upregulated. But then he added a second thing to rationalize why this is true. He said that it seems that cancer cells have defective mitochondria. That's why when the oxygen is there, they are not able to use oxygen. That's why glycolysis is upregulated. Is it making sense to all of you guys? Why did he say that? Because in 1940s, microscope was not that powerful, right, like it is today. So when he said this thing, for decades, people were saying, yeah, mitochondria are defective in cancer cells. Then in 80s, it came out that mitochondria are not defective in cancer cells at all. They are healthy, but they are still using a lot of glucose for the same reason. Glucose is providing starting material to build the rest of the cell, right? So this part is aerobic glycolysis. So now we know that cancer cells use aerobic glycolysis. So now we give inhibitors of aerobic glycolysis, cancer cells die. So basically, if you know what is going wrong, you can develop inhibitors of those pathways and you can target cancer cells. That's what this is showing. Okay. So this is again a similar thing. So cancer cells have these features. So based on these features, you can have the inhibitors to shut these pathways down. That's what it is, right? Don't worry about the details of everything, okay? Because if we talk about the details, it will become too intense there. We have to get the general idea, and I will take you in details where they are necessary, okay? But not all details are necessary here. But the good news is that we know so many features of cancer, and we have developed enormous amount of drugs, okay, to treat cancer in many different ways. Cancer patients did not survive before, right? They had very, very low life expectancy, right? But now it is really treatable, okay? In our department, there is a student who had cancer and he's completely free of cancer. I had an undergraduate in my lab, she had leukemia, completely free of leukemia. So we actually have made huge progress, okay? So now I will tell you immunotherapy. I'm sure you all are very interested in hearing about immunotherapy, okay? And almost every lecture will have some component of immunotherapy. But today I will tell you a very simple version of it. But I have lecture where I will explain exactly what is immune system. Because you must be wondering what is immune system and what cells are there and how exactly does it work and where is it located in your body, right? We know where the bones are, we know where the stomach is, but where is the immune system? Where is it hiding? Where is the location? What are the components? I'll tell you everything. But for today, again, introduction. So what happens is that in very, very simple words, we have many different kinds of cells which protect us. They all form part of the immune system. So one of the cells which are very famous are these T cells, okay? B cells and T cells, that it doesn't mean that they are the most important ones. All the cells are equally important. But what T cells do, they basically fight off foreign things in our body, okay? So what happens is that if our immune system is too active, that is also very bad. So suppose I am a normal person with no disease and something happened and my immune system is activated. So it is good if it is activated, but if it gets activated for too long, then it will start recognizing myself, all the self thing as foreign things, and it will kill me by autoimmunity. Like, you know, many autoimmune diseases are there where the immune system does not recognize your own body as your own body and it will start killing you. One of the very famous disease which occurs which is an autoimmune disease is myasthenia gravis. What happens? The immune system is recognizing self as foreign and it is destroying our own body, thinking it is a foreign thing, right? So the bottom line is that you have to shut down immune system also. So if T cells become active, good. They are defending. Too active, they will kill us. You need to shut it down. So body also has a way to shut down hyperactive T cells, okay? And tumor cells hijack that mechanism to fool the T cells, okay? So what happens is that T cells have a receptor, okay? That receptor is PD1, PD-1. So what is the function of that receptor? So imagine this is T cell and imagine this thumb is a receptor, PD1 receptor. So what happens is that, and this is very active, hyperactive, right? So this is APC cells, these are another kind of immune cells, and it will recognize that this T cell is very active. So what it will do, when you have a receptor binds ligand, right? So it will make PD ligand, so it is called PDL1, L stands for ligand. So this is PD1, this is PDL1. So this cell which has PDL1 will bind here. Now this receptor is engaged and the T cells will become quiescent. Okay, so this is our body's defense system to take hyperactive T cells and bring them back to resting stage. Okay, makes sense? Too active, we have to bring it like that. Okay, so like I said, you smell too much, our body shut down the smelling system. We cannot smell it sometime, right? This always happens. This is homeostasis. So this needs to be shut down. So now what tumor cells do, they make PD-L1. So when T cells come, they engage it in the same way, and T cells are off, and tumor cells survive. This is a very, very common mechanism to shut down the immune system. So that's what is shown here. So, so this is T cells, and imagine this is PD receptor, PD-1, and this tumor cell is making the ligand for PD-1, which is PD-L1, and now when it binds here, these T cells are off, immune system shuts down. So now we are giving therapy so that something else binds here, or we remove, we shut this down. Now that this, as long as these two don't bind, this is active. This something which binds here, this is not the same as this thing. So this binding does not shut off the T cells, and T cells will kill the tumor cells. I know of a case at present time, a person who was given this therapy and his cancer is stabilized and metastatic spots are disappearing. So this is very powerful therapy. Okay, so there are many ways to do immunotherapy. We'll discuss each of these in details later on. Okay, so these are some of the ways. Okay, so immune checkpoint inhibitors. So PD-1 and PD-L1. Okay, I'm going to show, you will probably hear many times this thing, PD-1 and PD-L1. Okay, so these are called immune checkpoint inhibitors because when immune system is too activated, it needs to be checked. That's why they're called immune checkpoint inhibitors. There are other guys too. Okay, then there is CAR T cell therapy. How many of you have heard of CAR T cell therapy? You have, right? It is one of the latest technology. And what you do, you actually make specialized T cells outside of the body. Then you give it back to the patient. So suppose somebody has cancer, you take out the T cells from that patient's body. Now you engineer them, then you put it back. These are like, you take out T cells and now you make them, you give them super weapon. Once they go, they killed tumor cells. Why we take out from the patient? Because if you give it back to that patient, there will be no immune rejection because it's from the same patient. But then you can also make some generic ones, and that can be given to any patient, like it is made in and kept at one place. Anyone who comes, you can give them. So we'll discuss this one also. Cancer vaccine. What is the principle of a vaccine? When we get a vaccine, what exactly is happening? Suppose we get a vaccine for coronavirus, right? What is a vaccine? So virus has this outer surface, right? So coronavirus, COVID virus has a spike protein. So you take part of the spike protein and you give an injection, just part of it, not the whole virus, just part of it. So when you give an injection in the body, the immune system will recognize as foreign. So it will make antibodies against this part of that small spike. Once the antibodies are there, so usually when we get vaccine, we get fever for a day or two. We are very sore. Why? Because the immune system is very active, right? Because it is fighting off, something has come. But we have given in such a small dose that it ends up with a fever, right? We don't get a disease. Now, if coronavirus hits that person, we already have antibodies. So we will clean up very quickly and we'll never get the disease. That's the principle of vaccine, any vaccine that we get, right? So cancer vaccine, we know this person has this tumor. If you take very, very, very small part of that component and you give it to the person, body will launch a huge immune response. And now all the antibodies are there, they will find the cancer cells, make it disappear. That's cancer vaccine. Okay, and then Same principle that you can make viruses which are engineered to selectively target cancer cells. So they will not touch normal cells, but they will make cancer cells disappear. So these are oncolytic viruses. And this is very interesting. I will tell you about these T cell engaging bispecific antibodies. So this bring T cells into close proximity to cancer cells, lead to this thing. So see, I am here today, right? And you guys are all here today. So we are interacting, right, in person. But this is not a very good example because this is probably live telecast and other people are hearing it and we are not in the same room and we are not interacting. But imagine the cancer is localized here, right? And T cells are roaming around. But if you make an antibody that one end is binding here, one end is binding here, now they are in close proximity, physically close proximity. And if you bring two things right in front of each other, these T cells will kill cancer cells. So antibodies have two arms. One is binding cancer cells, one is binding T cell. Close together, T cells will kill cancer cells. Remember, T cells are immune system cells which destroy foreign things. Cancer is foreign, right? It is right you bring the army right in front of the enemy and hopefully the enemy will be killed, right? Unless the enemy is smart enough to kill that. And cancer cells are smart, okay? I don't have those pictures, but it is amazing the kind of things they do. I mean, this is not a laughing matter, but if you see the real pictures, it is very funny. So cancer cells will make long projections, right? Go to the neighboring cell, long projection, like really sitting here, going all the way to you, and then stealing the mitochondria from that cell and bringing it. And people have taken pictures, mitochondria migrating from there to there. So they do all sorts of weird things, okay? But if you know what they are doing, you can treat it. If you don't know what they are doing, you cannot treat it. So finding what they're doing is very important, right? Okay, so what are immune checkpoints? So immune checkpoints are normal inhibitory molecules on immune cells that prevent autoimmunity, excessive immune activation, tissue damage, and cancer cells hijack these checkpoints to turn T cells off. So let me repeat this thing once again. You get vaccine, right? And most of the times we get vaccine, we are really down for a couple of days. We have fever, we have headache, we have all of these things. So that's when the immune system is becoming active, okay? And that is just very tiny activation of immune system. So when immune system is active for a long time, it can kill the body because it is just too active. So our human body have checkpoints. If immune cells become too active, they check it and bring it to normal level, okay, which is really, really required. We get fever for 1 or 2 days because this immune checkpoint is inhibiting the signaling, okay. So cancer cells will use this method to completely shut down immune system to survive, okay. So I'm coming back to PD-1 and PD-L1 again, okay. So please pay attention here, and I will show you this kind of slide many times again. PD-1 therapy is extremely effective in cancer. Okay, this is extremely effective in cancer, and this is one of the best therapy right now. So what happens is that PD-1 is a receptor. You know what is a receptor? So there is a cell. Receptors are expressed at the cell surface and they bind the ligand. So this is the ligand. They bind the ligand and the signaling is turned on. Sometimes binding the ligand can inhibit them. Sometimes it can turn it on. So here is a T cell. Don't pay attention to too much information here, but just look at this. This is a T cell and this is an antigen presenting cell. These are normal cells, okay? So what happens is that, this PD-1 receptor is found on T cells, B cell, NK cells, myeloid cells. These are all immune cells. Again, later on I'll tell you what the functions are, but these are all immune cells. So this is present there, and so many cells, they have the opposite of PD-1. So this is PD-1, they have PD-L1. This is the ligand which will bind here. So suppose this is APC cell, different cell, this is a different cell. This is my hand, this hand is T cell, this hand is APC cells, antigen presenting cell. This hand, this is PD1 ligand, this is PD1, this is PDL1, the ligand, right? This cell is T cell, this is APC cells. So what happens if they come together like this? I'm shutting this guy down by just binding it here. I'm shutting it down. So that's what is shown here. So T cells express PD1, antigen presenting cells express PDL1, and when they bind, this is shut off. Okay? And that is a normal mechanism in all our bodies to keep immune system in check. It's not too activated. So what cancer cells will do, cancer cells are not supposed to have PDL1, but they will express PDL1. So when T cells come to kill cancer cells, they lock it. And once they lock it, T cells are useless and they cannot kill cancer cells. Make sense? So if you give a drug, it's a very expensive drug if you give, because you are giving actually PDL1 to the patient. Okay, it's not a small molecule, it's not aspirin, it is actually a protein you are giving, the whole protein. Okay, so it's a very expensive therapy, but it is a very common therapy these days. You give something which will inhibit this interaction, then immune system will remain active, T cells will kill tumor cells. Okay? So PD1 and PDL1 are normal checkpoint, which is in normal cells essential for preventing autoimmunity, controlling inflammation, protecting sensitive organs. So yeah, go ahead.
Is there any difference between the PDL1 and the APCs and the cancer cell? Is there any difference?
So they still bind. So even if there are enough differences, they still bind. So there is a possibility. I have never checked that thing. So it is possible that there are some differences, but they still bind to these T cells. And it is possible that there are some mutations in cancer cells so that it binds even more tightly. And if it binds even more tightly, it's even worse because this guy is going to be shut off completely. So, but very good question. I don't think, at least in my knowledge, I haven't read anywhere that this is more effective. But based on my experience, what cancer cells do, I would imagine that they have even tighter binding there so that they can completely shut it off. Okay. So cancer cells exploit them, but their original purpose is protective. So our body has this system to keep us safe, but cancer cells has hijacked it for their own good. Okay? It is like you have a power, but it goes to the wrong person, it becomes abusive power, right? That's what it is. So this is a very beautiful picture. So this is showing PD1 ligand. So this is the ligand, right? This Y-shaped is, this is the receptor, not the ligand. This is the receptor on a normal T cell. Tumor cells express the ligand which binds the receptor, and when it binds, it is inhibited. Okay? And if you add anti-PDL1 or if you add anti-PD1, you inhibit this, they cannot talk to each other, right? You either inhibit this or you inhibit this. Okay? Now, they cannot talk to each other. T cell remains active. It works. It works miraculously in patient. It is a systemic therapy, but it is a very powerful therapy. I mean, it has saved the lives of millions of people. It is that effective. Okay? So this is just a cartoon showing these tumor cells. They will bind, and T cells, they go to sleep. Yes?
Does this immune therapy only limited to leukemia? Because T cells have a low invasion power to solid tumors.
No. T cell, it is applicable to every cancer, most of the cancers. So like I was telling you outside, it is very effective in solid tumors. It is very effective in solid tumors. And if you have low T cells, you can also use engineered T cells from outside. You can weaponize T cells, make them even more powerful, and you can give them T cells from outside, and then they can fight it.
Actually, right now it is very evident that scientists are shifting towards CAR-M therapy because CAR-T doesn't work on solid tumors. So they are producing CAR-M to treat solid tumors because
we will come to CAR-T, CAR-M, all the things. We will come to NK therapy, CAR-NK, all of those things. Okay, and I will tell you the advantages, disadvantages of all of those things as we go along. But these are really the emerging areas of cancer therapy. So this is just to say that cancer cells hijack this normal pathway. Tumor cell upregulate PD-L1 to mimic healthy tissue ligands. Don't attack me, I'm your guy, right? And that's how they shut down T cells. And this creates immune evasion. This is yet another picture of the same thing. So you knows now. And if you add the drug, now T cells will attack tumor cells and tumor cells will disappear. I have seen a real case where I've been monitoring a patient and immunotherapy has made metastatic lesions disappeared. Like they disappear slowly. So this is very powerful therapy. Okay, this is once again. Okay, I want to say one more thing. Go ahead.
The anti-PD-L1 drug would affect the natural PD-L1 too, then it will not it generate an autoimmune disease or something?
So, it is a very good question. So, what happens in cancer? Cancer is a fatal disease, right? So, cancer therapy is, it's not good for normal cells. So, immunotherapy is also considered a toxic therapy because it's systemic, it is exposed to normal cells. So, immunotherapy also is toxic eventually. But when you are fighting cancer, it's a question of survival there, right? So it's like, so it is affecting normal cells. And then, the sad thing is that the cancer cells develop resistance to this also. So I want to give a lot of information to you so that you can put your minds together to find even better therapies, all of you, in future, right? You have to know the field before you can design something, right? So I want to tell you about something else also. So one of the most famous immunotherapy is PD-1, PD-L1. You either inhibit PD-1 or PD-L1, same effect, right? But there is another very powerful therapy, which is targeting CTLA4. Okay, this is yet another kind of therapy, exactly like PD-1, PD-L1. Okay, so in this one, these T cells, they express a receptor called CTLA4 receptor. Okay, so when they express this thing, this antigen presenting cells, they express B7, they bind there and they inhibit it. Okay, so what cancer cells do? Cancer cells do not express B7. Here they are using a different trick. Like suppose there is a cancer cell, so they cannot directly attack T cells, but they know that these cells can attack T cells, so they will express these cells more, so that these cells come and inhibit it. And so there is a doctor in Max Hospital in Delhi, right? He's an oncologist. So he was talking to a patient and he said that he has found that CTLA4 therapy is usually more effective in patients than PD-L1 or PD-1. So, but that is his experience. So he said it is better to give PD-L1, PDA because it's less toxic to the patients. Like you said, toxicity is always an issue. But he said if it doesn't work, in his experience, this therapy is even better than that. So this is yet another way to treat, to activate the immune system. Okay, so this has too much information. Maybe I will skip this slide here because you guys are in the introductory stage. But what this slide is telling you basically that it is like, I cannot kill my enemy, so I bring in the army to kill the enemy. So the tumor cell is bringing in other cells which will kill the CTLA receptor of T cells, to kill T cells. So this is telling you a lot of that. They will recruit Treg cells, and Treg cells will bind this thing, and basically APC will inhibit it. So they will bring, it is too many ways to basically keep the immune system shut down. Don't worry about all that information. It will be too much information at this stage, okay, when we are still at the introductory stage. But you will have all of these things in your handout, so you can read it. If you have questions, ask me, okay? I know many of you are waiting for CAR T cell therapy. So what exactly is CAR T cell therapy? So this is obviously a T cell therapy. So this is a very, so suppose you know that the enemy is very powerful and our defense system is the army, right, which will kill the enemy. So you train the army, you give them weapons so that they are more, and you really make them very powerful so that they can kill the enemies, right? That is what CAR T therapy is. So what you do, you take the patient, you remove the T cells from the patient. If you remove it from the patient and give it back to the patient, there will be no immune rejection, right? So that is safer. So that's why they use the T cells from the patient. And then what they do, they change the DNA of the T cells so that it accepts special receptors. These are called chimeric antigen receptor. That's where the CAR comes from. And these receptors are synthesized at the cell surface in such a way that they turn on the power of T cells, and T cells become very powerful. And then you grow millions of these T cells, you give it back to the patient, and that is very effective in killing the tumor cells. So this one has slightly more information. So that was a generic one. So what you do, you take out the T cells, you engineer the T cells by recombinant DNA technology, and you add the DNA so that they express these receptors. They look like these kind of receptors, okay? These are chimeric antigen receptor. That's why this word CAR comes from. And now you inject it back to the patient, and when you inject it, it activates the immune system, killing the tumor here. So this is a very powerful, these are very expensive therapies. And keep in mind, many of you are chemists, right? So if you give a drug which is a small molecule, right? many drugs like Gleevec is a small molecule. That is very cheap because it's orally available drug, right? But these are biologics, like a whole this thing. So they are very unstable also, right? You make proteins, it can denature very quickly. So these are also very hard to make. If you make your compound, every single time you can make it the same purity, right? Chemistry is like this, but when you make proteins, it uses its own way, and there is batch to batch to batch variability. So it is harder to make, more expensive to make, but it is very, very effective. Okay, and we will spend a lot of time I will tell you all the things about that. So now we look at cancer vaccines. I already told you what a vaccine is. So what is a vaccine? Just to remind you that suppose a virus attacks the immune system, right? And the immune system will react. So what we do, we train the immune system by giving just the speck of that virus. Speck, so that speck cannot cause the disease, but it will actually activate the immune system, right? So when the immune system is activated, it will make antibodies. Once it makes the antibodies, our body is so smart. Any antibody it ever made, some part of those antibodies are stored for as long as we are alive. Okay, and anytime that particular thing comes, those antibodies are already there, and they will very rapidly divide and launch the attack very quickly. Okay, so that's what it is. So you train the system, and now, this is what we do when we get so many vaccines. Even the newborn babies, they get so many vaccines. You're training the baby to have all of those antibodies, right? So you can do the same thing with cancer cells. You can find what are the specific things for the cancer cells. What is the advantage? The things you are picking up are not picked up by, are not present in normal cells. So it will only attack the cancer cells and it will leave the normal cells, right? So those are the cancer vaccine, and once the immune system is active, cancer cells will die. So this is cancer cells from a patient are killed and in some case broken into small parts. Why killed? You don't want to put live cells. If you're putting live cells, they will cause cancer. That's really bad. So you really break them apart. You take the proteins which are expressed there, specifically there. You put it back, you start the immune reaction, you make the vaccine, you give it to the patient, and it will kill the tumor cells. Okay, so this is even more details exactly how you take it out, okay, and how you can give it back to the patient. Now we come to oncolytic viruses. So now you make a special virus, okay, but you change the DNA. So you make so you don't want to give a virus which will create a new disease in the human body. So you remove the disease causing part so that it is a safe virus, and then you insert new DNA which will stimulate the immune system. And then you give that virus, immune system is activated and cancer cells will die. That's the whole idea. So when you give oncolytic virus, so you give it, it will not affect the normal cells, but it will affect the cancer cells. It will replicate in the cancer cells and kill the cancer cells. Okay? So the tumor lyses and it is just completely gone here. Okay? All right. So now we come to T cell engaging bispecific antibodies. So these kind of things have been really very effective. This has revolutionized treatment for certain cancer. It has a potential for long-term remission and reduced side effects as compared to chemotherapy. Okay, so this is that you bring the two guys, it's like somebody's culprit is there and you bring the police right next to that. So if you bring that right person next to it, we are taking care of the problem, right? So that's what you're doing. You are making an antibody which will bind to an immune cell and the tumor cells simultaneously. So this gets activated, it will attack this by releasing, by different mechanisms. Here it is releasing perforin granzyme. So what it does, it makes holes in it, goes inside, kills the cell. Okay? It's like you bring the police and the police start shooting. The enemy is gone. Okay? So this is another, you bring the antibodies here, and they come next to each other, they engage, and now here it is releasing cytokines which will kill the tumor cells. All right? What time? Is it done? Okay. Okay, I will stop here. All right?
Thank you, ma'am, for the wonderful talk. Now we'll proceed for the lunch. So I will request all the participants to come back at 2 p.m. for the next talk. Thank you.
So it's 1 p.m. now.
Yes. So we'll get back at 2.
Yeah, okay. All right.
Welcome back, everyone. Now I would like to request ma'am to continue his lecture. Thank you.
So this is still part of lecture 2, so I will go a little bit quickly to finish it off, and then we'll go to lecture 3. So this is just a difference between chemotherapy, targeted therapy and immunotherapy. Okay? So chemotherapy, so anytime you hear chemotherapy, so what exactly does it mean? So chemotherapy is based on a simple principle that cancer cells divide very rapidly, right? Other cells are also dividing in our body, but not as fast as cancer cells. So if you target those pathways which are involved in cell division, cancer cells will die. But what is the side effect? Normal cells will also die, especially those which are dividing very quickly. So it targets dividing cells, but the side effects are hair loss, intestinal damage, nausea. Remember I told you that our stomach cells, intestinal cells, they are involved in actual churning, right? So these cells are very rapidly dividing to replenish and make new cells, so the damaged cells are removed. So when you target division, cell division, these cells are affected the most, which results in nausea and hair loss and intestinal damage. So, and then the worst part is that this is the worst part of cancer. You develop a very good therapy and cancer cells will eventually develop resistance towards them. They mutate, they change themselves so that they no longer are affected by that therapy. Targeted therapy is that you find the culprit in the cancer cell and then you target that thing. And if that particular thing is not present in normal cells, then it is specific to cancer cells. So you have relatively less toxicity, okay, because you are targeting only the cancer cells. But usually it is hard to find something which is only present in cancer cells. Usually what happens, it is also present in normal cells, but a lot more in cancer cells. So cancer cells die, but these cells are also affected because they have the same pathway. Okay, so liver problems, diarrhea, skin rash, these are the common things. They also develop resistance. Immunotherapy is very expensive. Okay, that is the side effect. The good news is that it is very effective. During lunch, we were talking about real case scenario where people have seen and they share that how effective is immunotherapy. So it is expensive, but it is very effective. Okay. But if you play with the immune system too much, it can lead to autoimmune disease, like you guys can think, right? You don't want immune system to be hyperactive all the time, right? So that's the side effect of that. Okay, so now let's go to cellular therapy. I briefly told you about this thing. So cellular therapy is a transplantation of human cells to replace or repair damaged tissue or cells. So remember somebody said that in leukemia, right, T cells can be affected, or in some cancers you have very few T cells left. Okay, so in that case you can take the T cells from them and then outside the body you take them out, and outside the body you replicate them just like you replicate the cells in a tissue culture plate. So you can make more and more and more T cells, and then you give it back to the patient, and you can keep on continuing this cycle, so to replenish T cells, right? So that is cellular therapy. Gene therapy, as the name suggests, that you can actually, suppose tumor suppressor is mutated and it's no longer working, you can put the tumor suppressor back in the cell, okay? Or if it is a defective gene, you can actually fix the gene and put it back there. There are many diseases apart from cancer which are also affected by defective gene. So cystic fibrosis is one of those diseases, right? It is very common in Western world. So there the chloride channel is not formed, and 30 years ago, cystic fibrosis patient, they had a median age of 12 years only, right? Right now it is about 40-50 years. So we have made huge progress in the field. So there you fix the defect of that chloride channel, and then you give it back to this patient, and you have basically cured the disease. So that is gene therapy. Okay, so in gene therapy, there are many ways to give the DNA back to the cell. Do you guys know what is a plasmid? Some of you know what is a plasmid, right? So plasmid is like this is a piece of DNA, and you add your DNA of interest here. Now this whole system, if you put it in a cell, it will make more DNA, DNA will make RNA, RNA will make protein, right? And you can have different kinds of vectors basically. Don't worry about the details. The point here is that you can get the DNA across in the cell, and it will make the required protein. So you can correct the disease-causing gene, you can put it back in the patient's body, and then basically you have fixed the disease. That's the gene therapy. Nanotechnology is just a way to deliver it. It's a delivery machine. So, you know, think about cancer, some sort of cancer. So one thing is that you developed a therapy which will only affect cancer cells. So you can give the pill to a person or you can inject something in the person and it's not going to affect the normal cells at all, right? And it will only kill the cancer cells. That is like a dream drug, right? Or we can do one thing, that the drug will affect normal cells to some extent. So we then trick the drug to go only to the tumor cells and normal cells don't take it, right? So what happens is that in many cases, these tumor cells, they express cell surface proteins which are not present in normal cells. So suppose this is my drug. So what I do, I attach a ligand which binds to the receptor in cancer cells. And these receptors are not present in normal cells. So my drug is only taken by cancer cells. Is it making sense to you guys? So let me repeat this thing here. So there is a receptor called folate receptor. So cancer cells have huge amount of folate receptors. Okay, and these are present in very, very, very small amount in normal cells. Okay, so what you do, these folate receptor binds a ligand called folate. Okay, so if you just give the folate, it is not good for cancer, right? It doesn't help. But now what you have to do, you have to deliver the drug only to cancer cells. So what you do, this is your drug. Okay, this is your drug. So instead of giving it in a pill form where it is all over the body, you attach folate to this drug. Okay, you attach folate to this drug, and when you give this, these cells will bind this folate and this drug will be taken inside the body, inside those cells. So now what you're doing, you're specifically delivering the drug. You don't care about folate. Folate is just a carrier. But the drug itself is going to the tumor cells. Make sense? So similarly, nanotechnology, what you do, you package the drug inside a nanoparticle. So it is like completely protected, right? And then you attach folate on the nanoparticle. So it will go and bind to the receptor. It's taken inside. So nanotechnology is very good for drug delivery. Nanoparticles are not the drug themselves. They can be the drug themselves. I will give you examples of that, but usually they are delivery vehicles for the drugs. Okay, so this is a nanoparticle. So we can attach the drug like a small molecule drug. We can attach an antibody. Antibody will bind that cancer cell only. We can attach DNA aptamer. Do you know what is a DNA aptamer? DNA aptamer is a piece of DNA which will form a special secondary structure, a tertiary structure. So it will not follow the double helical DNA, but it will form all this kind of tertiary structure, and then it will bind only to cancer cells. So you have to make a DNA aptamer which is specific for cancer cells. So the idea is that you are delivering the drug to the right address. It's not randomly going everywhere in the body. It is being delivered to the cancer cells. Okay. So here, targeting peptide, same thing, deliver it to the cancer cells. And you can fill this with drugs, or what you can do, you can add imaging agent like fluorescent molecules. Some of you, I'm assuming, work on fluorescent molecules, right? So when you work on fluorescent molecules, you can attach fluorescent molecule. What is the advantage? Now cancer will light up, right? So what people have done, they have taken this folate and attached to fluorescent molecules. I think Professor Venkatesh has also done this work during his postdoc. So when you do that, then these, wherever cancer is expressed, usually they have high levels of folate receptor. So that will go and light up the cancer. So now it is FDA approved. You can inject the drug into the patient and then do the surgery. So if there are any cancer cells left which cannot be seen under the microscope, the fluorescent signal is very strong, you can actually remove every single cell during surgery because it lights up, right? So you can do a lot of things using nanoparticles, okay? So conventional is that you give the drug by injection or you give that, and there is a lot of toxicity because it's going everywhere. But using nanoparticles, you can deliver it right to the spot, so that it will be less systemic toxicity. Make sense? All right, so let's talk about traditional medicine versus precision medicine. So in very simple terms, traditional medicine means you give chemotherapy or anything and it is going everywhere in our body, okay, whether it is chemotherapy or radiation. But precision medicine is that you actually find the problem in that person's cancer, the exact problem. What are the things which went wrong in that person? And then you make a tailored therapy for that person. Okay, it is not traditional chemotherapy, it is not even traditional immunotherapy, but it is specific for that particular cancer. That is precision, and it can be highly precised, right? Extremely precised. So that is precision. So you don't make one generic drug and you give it to all of them. No, you give specific drug to specific patients. That is precision. Okay, so here there is one treatment and you give the same drug to all the cancer patients. So some of them respond, others they don't respond. Okay, but precision, you actually do genetic profiling, proteomics, metabolomics, all of these things, and you really understand what's wrong with this particular person versus this one, and then accordingly you develop treatments. Okay, so that is precision. So for precision medicine, you have to have accurate diagnosis, right? You have to really figure out what went wrong in this particular cancer. Okay, and then you do biopsy, you isolate it, you do everything. Then you can even use animal model and figure out what drugs are going to be most effective. Okay, then you give targeted treatment and then it will work, and eventually that will lead to much higher health outcome. So for example, this is just one step towards precision medicine. There is a test called Oncotype, Oncotype X or something like that. I will give you the exact name tomorrow. Some onco kind of test. So there are 21 genes they test for breast cancer. And why they test these 21 genes? Because these 21 genes are often modified in cancer, in breast cancer. So once they check it, then they know, okay, this person has this protein which is abnormal, and then they give the drug against that protein. That is precision medicine. Okay, so so that's what they are showing here, that now people use AI in everything, right? So AI can so what is the limitation of AI? So imagine a scenario where all the data from all the countries, from all the patients are fed into AI, how they responded, what was the diagnosis, how they were treated, what was the response rate. The more data you feed into AI, the better outcome it will provide you, right? Because it is analyzing all the patient. Human mind cannot analyze so many possibilities, right? When there are so many, such diversity, so many treatments, such different kinds of cancer. So AI can really help, and we will discuss the AI thing at the very end also. So precision medicine, we are hoping that AI will make a huge impact in diagnosing the disease and really making tailored medicines for in each and every case. The other thing is that drug repurposing. So, so when you make a drug for any disease, right, it takes a long time. It takes a long time, and usually it takes billions of dollars to make one drug. If you synthesize in your lab, it may take $10 or 10,000 rupees, or maybe at the most 1 lakh rupees to make huge amount of this thing. And if you make a pill, the pill will actually probably cost you 100 rupees, right? Or even less than 100 rupees, right? But to identify that pill, you have spent billions of dollars to make sure, because a huge amount of research goes in every single drug, right? So it is very common to spend billions of dollars to find one drug for this thing. But then many times you realize that this drug for diabetes is actually very good cancer drug. Okay, so once you realize that, then you give that drug to cancer patients. Now the beauty is that you have done already so much clinical trial on that drug already for diabetes. You know it is not toxic to the patient if it is given in this amount. So now if you have to repurpose it for cancer, it's a very fast process because it's already FDA approved drug. And I will give you a real example. Have you guys heard of this drug called metformin? Many of you have heard, right? It is #1 diabetes medicine. It is also given to pre-diabetic patient, right, to prevent diabetes, right? And metformin is an excellent anti-cancer drug. This is one of those drugs which has better side effect. You take it for diabetes and you don't get cancer. Wonderful, right? That is the side effect, no cancer in return. And it has actually no side effect. If you take it, is really good drug. So that is drug repurposing. So if you can use an approved drug to fix something else, that's really good. So that's what it is saying. If you repurpose a approved drug, it takes a lot less time, lot less money. Okay, so that was the main message here. Now, do you guys know what are antibodies? Some of you know, all of you know, right? So antibodies, Lake, Nath, do you know what are monoclonal antibodies? You don't know. You were not responding. Please ask me this thing. So monoclonal antibodies, so have you, so when we get infected, right, our body makes antibodies to bind those infected particles, and once they bind, they destroy it, right? So antibodies binds their target with extreme high affinity, really, really tight affinity. Okay, so that's where antibodies started. So suppose you have cancer cells, and cancer cells have the receptors, like these receptors which bind the ligand and turn on the cancer signaling. So if you make antibodies which will bind these receptors, okay, so they are basically closing the receptor and nothing will bind there anymore. So antibodies happen to be Y-shaped like this. They are this kind of shape, and this is the part where they bind their target stuff. Okay, this is a very generic, simple explanation. So why did I say antibody? So suppose I ask you, to any of you, to make a small molecule which will bind the target very tightly. Sometimes it is easy, sometimes it is very hard. The second thing about small molecules is that even if you make something which will bind it very tightly, the chances are it will also bind something else very tightly. So they usually are not monospecific. They inhibit many targets at once. This is the limitation of small molecules. Antibodies, the biggest advantage of antibodies is that they are extremely specific. See, our body recognizes the antigen, the thing they bind, with 100% specificity. So if you make small change in that, antibodies will not recognize. So it's very hard to make the antibody. They are huge, everything, but they're 100% specific for their target. So side effect is less, right? So that's what it is saying. And now it is becoming very easy to make monoclonal antibodies, and it is actually used in patients. Okay, so anytime you use a small molecule, it is cheaper, easier to administer. Anytime you use monoclonal antibodies, those of you who run protein gels, right? Usually if you have to make a protein which is 50-60 kilodalton, if you have to make a 20 kilodalton protein, it's a lot easier than 50-60. If you have to make 100 kilodalton, it is even harder. Antibodies are even bigger than that. So it's pretty expensive to make them. It is not that expensive to make them. It is very hard to keep them in the active form. They denature over time. So imagine you make a drug and it gets denatured in the pharmacy store. So that is the hard part with biologics like proteins and antibodies and all of these things, unlike small molecules which are much more stable. But they are still used, okay? So look at this thing, this is really interesting. Small molecules like aspirin, it is made of 21 atoms. Okay, so it is like if you think of small molecule, if it is the size of a bike, then antibodies, they are like a plane. They are big. So look at this thing, 21 atoms, large molecular drug would have 3,000 atoms, antibody has 25,000 atoms. It's really huge. Okay, so small molecular drug, even if you make a big molecule drug, you go from bike to car, but this is like the really big Airbus, 380 Airbus. Have you seen the Airbus? It is no longer working because it consumes too much fuel, right? So now they use Dreamliner and other planes. But Airbus, if you take the Airbus, it is the most steady plane. It it doesn't go with the turbulence like we have all experienced. You never experience turbulence in those planes. They are very steady, very nice planes and very beautiful looking planes. Okay, so imagine a bike versus those kind of planes. So these are much bigger. These are called biologics because these are biological molecules, right? So they are called biologics. So this is what small molecules, what are the advantage? Like aspirin, they can go inside the cell very easily. So they penetrate the cell membranes and target intracellular proteins. Most of the targets are intracellular, but you can have extracellular also. Generally more cost effective, can be administered orally like we take the medicine. May have off-target effects due to binding to multiple proteins. This is the side effect. Usually small molecules are not 100% specific. They target many molecules. So suppose I am targeting, this is culprit here, but it is hitting a lot of normal cells also. So small molecules do not target one target. They can target multiple because they are not that specific. But if you compare this thing, they are extremely specific. They will only target that one thing. But they can trigger immune response. They are so big that the immune system will become active. Okay, this is given by injection. The good news is that they have longer life. So if you give immunotherapy, you have to give every 3 to 4 weeks. So they have longer life in the body. You can actually, those of you who work with protein engineering, it's very easy to keep changing the protein of interest. And they can be used to deliver payloads. But the bad news is that they are so big, they don't cross the cell membrane. So if your culprit is extracellular, like a receptor, it works very beautifully. But if it is inside the cell, it will not work. But then we have a solution to that also. I'll tell you later about those solutions. Okay. So this is a normal way. So so what we do in our lab, that we identify the culprits, the real culprits in cancer and Alzheimer's disease. So once you identify the culprit, yeah, I know this protein is the culprit, then what do you do next? You make a small molecule against that protein or an antibody against that protein, and you basically inhibit that protein or kill that protein or degrade that protein, right? So that's what it is saying, that first you identify the target, what is wrong. Once you identify it, then you make the compound. So you know what pharma industries do? They have millions of compound library. So instead of doing chemistry one at a time, they take those millions of compounds, they screen against that target. Once they find the hit, they do SAR with all the related compounds. Once they find the hit, then they do modeling. Then they say, oh, this compound is binding like this, let's put a methyl group there or put something there. It should bind better, right? So then they optimize that molecule and then they find a very powerful, very potent molecule. That's the way. So that's what it is. They do the lead generation. Once they get the first, then they do optimization to make it more and more potent. So very focused chemistry. And then it goes in animal models and clinical trials. Okay. So now let's say how we make antibodies. Has anyone heard of antibodies formation, any idea? Probably not, right? It's not a very common technique everywhere. Some people do it, not a lot of people. So the easy way, do you guys use antibodies in your lab? Anyone? You are using antibodies, so we are buying it from the market, right? But suppose you work with a protein for which you don't have antibody. These days you find antibodies towards anything, but it wasn't that before, right? So I have also made antibodies when I was a postdoc. So what you do, you immunize the animal with an antigen. So suppose in my case it was PDGFR antibody. So what we did, we took a part of PDGF receptor, PDGFR, and we injected it. I mean, I did not. We sent it to a company. They injected it in mice or rabbit. In my case, it was rabbit. So the rabbit had an immune response. Rabbit made the antibody. We isolated the antibody, or basically the company isolated the antibody. They purified the antibody. They sent it to us. Okay. So that's what they are. So the antibodies are produced, you isolate it, and then you can directly use it, or you can make a cell line out of that antibody. So you can fuse it to myeloma, and you can then make cells which will always produce antibodies for you. You never have to go to the animal. But it is a long process. So in our case, we just got the blood sera from the rabbit and used it, and it worked beautifully. But people also make the cell line out of it. So it's a long process, but making antibody is not hard. What you have a constant supply. You don't have to synthesize it again because it will make it for you. Purifying it is one day's work, so it's very, very easy. The disadvantage is that keeping it active, because it denatures very quickly. Those of you who make proteins, you know it will get denatured. You cannot leave it at room temperature or anything, right? So that's what it is. So you diagnose the problem, you identify the target, and once you identify the target, then there are many different ways to target that protein of interest. Okay? So the other thing is that antibodies are very, very big, right? How do you deliver it? So this is a problem with biologics. We have the thing, but it will not go inside the cell. So now the delivery becomes a problem. Okay? So this is just many different ways. You can do nanoparticles, you can, and also what happens is that when you inject it, even when you inject it, they may degrade it in the blood. They may have a very short half-life in the blood. So you have to also protect it from the blood. It is possible it will go in the liver will clean it out of the system. So you have to fool the rest of the body so that it has a long half-life, but you sometimes don't want a very long half-life because if it stays for very long, it may be toxic. So there are many considerations there, okay? All right. So we finished the second part.